Good morning, good afternoon, or good evening, or good day, good next day to those of you in other countries. We would like to extend a welcome to you to this webinar. My name is Ann Brazo. I'm the founder of MPN Advocacy and Education International. We are celebrating our soon-to-be 14th year, I believe, in September. And prior to this, I was with the MPN Research Foundation. So I've been around for a long time, if you don't know that. You can see the roadmaps on my face. Anyway, we are so excited today because we try to bring to you at least once a year clinical trial updates from our partners, from our biopharma partners. And these presentations are so critical and so important to you as a patient or caregiver, and even as a physician and an MPN specialist or hematologist that sees MPN patients, because you will learn what's going on at each of these companies, what drugs are in the pipeline, what's happening in these clinical trials. And it is extraordinarily important. Since I have been in this space, there are 4 new drugs in the market, or not too new, some of them, but there was nothing for MPN patients when I started. So this is pretty remarkable to me and to many of us. So without further ado, I'd like to introduce our first presenter, who is from Incyte. And Incyte has been a partner with us for years. They have supported us through many tough times, and we're very happy to have Vakal Bahat, who is the Senior Scientific Director of U.S. Medical Affairs. Please welcome Dr. Bahat. Oops.
Can you hear me?
Yes.
Okay, perfect. Hi, everyone. A pleasure to be participating today, and thank you so much for the opportunity to share our current pipeline in MPN clinical trials, and all the work that we have been doing within MPNs from a clinical development perspective. As Anne really stated nicely, I am one of the scientific directors within U.S. Medical Affairs at Incyte. I'm a pharmacist by background. I worked in clinical practice for many, many years prior to joining Incyte, taking care of patients with hematologic malignancies like MPNs. Being a part of this organization has just been so valuable and so heartwarming for me to be able to work with clinicians and scientists to help move the science forward. And I'm so happy to be able to share some of those key highlights of what we've been up to over the past several years with you all. So at this point, I'll try to share my screen. Hopefully, the share's okay. Okay, I hope you all can see my screen with the blue slide, if that's okay.
Yes, it's
Okay,
there.
perfect. So with regards to, just a few disclaimers, all of these agents and our pipeline program that we are studying, that I will go over in the next 5 minutes or so, is really, it's in clinical development. As you all know, that these agents are not approved. So the efficacy and safety of these compounds still needs to be further validated, and we're working hard to study those in the right clinical settings with patients with MPN. And there's no guarantee that these agents will be commercially available. And any of the data that I present today does not imply clinical benefit, but just shows sort of the trends in what we are seeing in the initially treated patients with MPNs. So just providing a historical perspective, Incyte has been really dedicated to MPNs for many, many years. And you could see our key agent, ruxolitinib, was approved in 2011 for patients with intermediate or high-risk myelofibrosis, followed by our subsequent approval in polycythemia vera in patients who have had an inadequate response to hydroxyurea or are intolerant to that therapy. Subsequently, ruxolitinib was also approved in the treatment of treatment refractory acute and chronic graft versus host disease. So the impact of this treatment has had far-reaching benefits to patients outside of just myelofibrosis and polycythemia vera. And we're very excited that this mechanism and this agent has provided benefit for so many patients living with MPNs, as well as outside of MPNs in acute and chronic graft versus host disease after bone marrow transplantation. This goes into the details of what our pipeline looks like within MPNs and GVHD. And I mentioned to you and I'll focus on MPNs here, is Jakafi, as you know, is approved in myelofibrosis and polycythemia vera, as I alluded to previously. But the three key pipeline agents and programs that I'll discuss with you today are highlighted in the blue box there. So we're investigating a novel BET inhibitor within myelofibrosis as monotherapy and as single agent alone, as well as in combination with ruxolitinib. So in efforts to improve those response rates that we are seeing with ruxolitinib alone in patients with myelofibrosis. We also have a novel targeted agent that targets a key specific mutation in a subset of patients with MF or ET with a monoclonal CALR, mutant CALR antibody. And this is also being investigated as single agent as well as hopes for studying in combination with ruxolitinib as well in the future to improve response rates in those patients who have a mutant CALR mutation, which I will go into a little bit more detail of how often that occurs in patients with MPNs. And then finally, we also have a very exciting target in targeting the specific driver of disease in MF and PV, which is the JAK2 V617F mutation. We're studying in the clinic an inhibitor that really targets JAK2 V617F in efforts to not only improve response rates in patients who have received ruxolitinib, but also providing a targeted approach to achieve potential disease modification in patients with MPNs and myelofibrosis. In this case, as we are studying in the initial patient populations. So I'll start out with our BET inhibitor first. So the BET inhibitor and BET proteins are really pathogenic in MPNs. They are epigenetic readers that really impact and regulate the growth and survival, as well as the inflammatory responses that occurs in patients with MPNs. When this agent was studied in mouse models, we showed that there was a reduction in inflammatory signals as well as reduction in bone marrow fibrosis. As you can tell, those preclinical studies really target the key drivers in patients who have MPNs. And furthermore, we showed that inhibition with combining JAK inhibition with agents such as ruxolitinib in combination with BET inhibitors can help to reduce the inflammatory cytokines that really drive the clinical manifestations of MPNs. So thinking about symptoms that patients may present with, as well as improving blood parameters. So improving blood counts, as well as improving spleen size. And then there was preliminary evidence in those mouse studies that showed that it reversed bone marrow fibrosis, which is a really exciting preclinical data that we are currently investigating in the clinic. And as you could see there, we have a phase 1 study that's ongoing that's evaluating an oral once-a-day BET inhibitor as not only single agent, but also in combination with ruxolitinib in patients with myelofibrosis in hopes of improving response rates in those patients who have potentially had suboptimal or are refractory to initial ruxolitinib therapy, and also in the setting of improving response rates in combination in achieving a higher depth of spleen volume reduction, as well as improving symptoms and blood cell parameters, such as improving white count, anemia improvement, as well as overall count improvement in these patients. So that study is ongoing and continuing to enroll patients. The next agent I'll go over is the very exciting mutant CALR inhibitor that we also have ongoing and is enrolling patients in the clinic. And mutant CALR is present in a subset of patients with MPNs, and mutations in the CALR protein can result in the activation of the TPO receptor, which leads to constituent activation of downstream JAK-STAT, which, as I alluded to previously, is one of the key drivers in MPNs. So in those subset of patients that have a mutant CALR mutations, this really provides that targeted approach to their disease without many off-target with the potential to limit any off-target toxicities. In the preclinical studies, and you could see there on the right-hand side, the targeting mutant CALR with a CALR antibody, it has been shown to reestablish reestablish the megakaryopoiesis, or improving counts in those in preclinical models, suggesting some disease modification activity in patients with mutated CALR. So now, based on those initial preclinical findings, there's 2 studies that are ongoing that are evaluating this antibody in patients with MF as well as ET, in investigating the further safety and efficacy and effectiveness of the mutant CALR antibody in patients with MPNs. And then finally, I'll provide an overview of the JAK2 V617F specific inhibitor that we have enrolling in that we also have available and enrolling patients in the clinic in phase 1 studies. You could see there that JAK2 V617F occurs in 65% of patients with myelofibrosis, and close to over 90% of patients with PV, and approximately half of the patients with ET. So as you can see, across the paradigm of MPNs, this is one of the most common mutations that is a key driver of the disease. What our V617F inhibitor does is that it really targets the pseudokinase domain that leads to that is responsible for constituent oncogenic signaling that leads to cell proliferation and the development of MPNs. So really targeting the key driver in the mutant clone that is the key driver of the disease. And this study is also this agent has also been studied in the preclinical setting in mouse models to have very selective and targeted approach to targeting the V617F mutation. So providing that next generation of agents beyond the JAK inhibitors that are available in standard of care in the clinic, it provides a novel and more targeted therapeutic option in targeting the most common mutation that occurs in patients across all the paradigms of MPNs in MF, PV, and ET. There's a phase 1 study that's ongoing and enrolling patients, and we're really excited to see clinical data over time on the safety and effectiveness of this agent in the clinic, building on the preclinical data. I thank you for your time. Those are the key areas where we are continuing to investigate within MPNs and show our dedication to MPNs through clinical research. And always thankful for this opportunity and thankful for all the patients participating on our trials that really help move these programs forward and generate really pivotal data. We work very closely with you as the MPN advocacy group as well as the patient community to help support these initiatives. And I just want to say thank you for all your support and in moving these programs and providing better treatment options for patients. If you have any additional questions, I've provided some of our contact information here, and happy to address any questions that you may have offline as well. So thank you very much for your time.
Thank you very much. This was fantastic information. I mean, there's so much hope out there for MPN patients currently that did not exist 22 years ago. I mean, it just wasn't there. And we have another presenter who has been around with me, pretty most of these years. But I will say that, and I applaud those also who are in clinical trials and who would like to be in clinical trials. There, you have this information now. Speak with your physician and see if you are a candidate, because this is just incredible news. So thank you very, very much for sharing with us.
Thank you for the opportunity.
Yes. So we're going to move on to Karyopharm Therapeutics and Travis Cooper, who's the principal medical science liaison, and global medical and global medical science, scientific affairs, sorry. And Travis, are you with us? Yes, you are.
Can you hear me?
Yes, I can.
Thanks,
And
Ian.
if you have shared your screen, and it looks like it'll be fine. So thank you for being with us, and we will eagerly listen to what you have to share.
Let me present mode. All right. Thank you so much, Ian. Can you still see that?
Nope, can't. Oh, there we are. Yes.
Okay, great. Well, thank you so much for allowing me to be here today. It's as you mentioned, Anne, my name is Travis Cooper. I'm a pharmacist by training, and I am part of the Global Medical and Scientific Affairs with Karyopharm, which may be a new name for many here in the audience. Thank you for allowing us to participate. We're a little bit new to MPNs. We're an established company in hematologic malignancies with our agent selinexor, an XPO1 inhibitor that I'll be discussing in detail today. A couple of things I want to talk about today is the Sentry trial, the Export MF034 phase 3 clinical trial. I'll give you a little bit of an update with some phase 1 data from that portion of that study. And then lastly, I'll briefly mention a phase 2 monotherapy study with selinexor, which is again, our XPO1 inhibitor that may be new to many here in the audience. So I'll give updates of that phase 1 as well as some of the phase 3 study that's ongoing and recruiting presently. So as mentioned before, the use of selinexor in myelofibrosis is truly investigational at this point, has not been approved by any regulatory agency or authority, and the safety and efficacy has not been established at this point. And so this information today will be a rationale for the ongoing recruiting trial for the Export 034, which is a combination study with ruxolitinib, as well as our 044 study that is a monotherapy study with selinexor. So let me briefly, tell you a little bit about Karyopharm. We are a small biopharma company that we are focused on really the discovery, development, and commercialization of a novel first-in-class agent that targets nuclear export and several related pathways, especially in MPNs, with the aim of treating both hematologic cancers as well as even solid tumors. So this is truly a pan-tumor agent. As I said, we're small, over 300 employees as of last year, 10 plus years in R&D. We've been around a while, headquartered in Newton, Massachusetts, and then we obviously have a footprint here in the U.S., but through our alliance partners, we have a global presence as well. So let me get into the meat of our discussion today. So this is our myelofibrosis clinical development program. This is the bulk of that. So we conducted initially a phase 1 study that has completed enrollment. This was a double-blind randomized trial of both 60 and 40 milligrams of selinexor plus ruxolitinib versus placebo plus ruxolitinib in JAK inhibitor treatment naive patients. And so what I'll do is I'll show you that phase 1 portion, which is a dose ranging study, as I mentioned, of 40 or 60 milligrams, and the results of that led into the phase 3. Those results were promising enough to actually move over the phase 2 portion and go directly into a phase 3, which I'll describe at the end. Also, lastly, I have it correct on this slide. The Export MF044 study is a phase 2 trial that is expanding that effort to evaluate selinexor in JAK inhibitor naive patients as a monotherapy option, especially in those patients with moderate thrombocythemia. And those 2 studies are, again, open and enrolling. So just briefly, I know this group knows the hallmarks and the issues and limitations we have in and unmet needs in myelofibrosis. We know that this hallmarks in myelofibrosis are really centered around aberrant blood and bone marrow differentiation, cytokine production that really leads to constitutional symptoms for patients, as well as ultimately bone marrow fibrosis and extramedullary hematopoiesis. And there is really a current and present unmet need beyond JAK inhibitors. We know that responses in terms of measured by SVR35 and TSS50, that about 50% of patients, or less than 50, excuse me, percent of patients achieve an SVR35 at week 24, which is our typical endpoint, regulatory endpoint in myelofibrosis. And so even if patients do achieve that durability is not long lasting. And so we know that probability of maintaining that spleen volume response decreases approximately by week 12. We also know that there's a need for disease modification, and that JAK inhibitors tend to have a limited ability to alter bone marrow fibrosis, inflammation, and prevent that disease progression. And so although there have been a number of key additions to the treatment landscape particularly with patients with cytopenias, there's still limited treatment options and may require suboptimal doses of those JAK inhibitors. And ultimately, we know that myelofibrosis is a persistent disease with leukemic transformation and decreased survival and significant morbidity in this disease state. So there is an unmet need here that we are trying to play a part in. And so let me tell you a little bit about selinexor. Selinexor is an XPO1 inhibitor, or exportin-1. I'll refer to that as XPO1 going forward. XPO1 is important in terms of nuclear to cytoplasmic transport of important cargo proteins, namely tumor suppressor proteins. And the one I've mentioned here is probably the most famous tumor suppressor protein, P53, important in terms of regulating gene expression and inducing apoptosis. And so we know that selinexor is approved in myeloma, as well as diffuse large B-cell lymphoma And we know that in terms of its mechanism of action, it induces p53-driven cell death by inhibiting XPO1 and maintaining p53 and other tumor suppressor proteins in the nucleus, so that it can actually do its job in terms of inducing apoptosis and cell cycle arrest. So how does that work into myelofibrosis and MPNs? Well, we know that the hallmark here is the JAK-STAT pathway and that aberrant signaling. And selinexor certainly has effects on the JAK-STAT pathway, but also outside of that JAK-STAT pathway, particularly with NF-kappa B. And certainly, I won't go through today, but we have a wealth of preclinical data that supports that in terms of these JAK-STAT as well as non-JAK-STAT pathways and synergy with JAK inhibitors. So let me move on to the phase 1 portion of our Export MF034. As I mentioned, this is in patients who are JAK inhibitor naive with either primary or secondary myelofibrosis. We did a dose-ranging study of 40 or 60 milligrams once weekly of selinexor as an oral drug, expanded that to 18 patients. And what I'll be talking about primarily here are the 14 patients in phase 1 who received a 60 milligram portion of that study. You can see some of the primary endpoints and secondary endpoints that I'll discuss today, particularly SVR35 and TSS50 at week 24. As I mentioned earlier, this led to the ongoing phase 3 study of selinexor, 60 milligrams weekly, plus ruxolitinib at standard dosing versus placebo plus ruxolitinib, and that is ongoing. A couple of the select inclusion criteria you can see below in terms of spleen volume. This included DIPS intermediate 1 who were symptomatic, intermediate 2 or high-risk patients, and these patients needed to have a platelet count over 100,000. So a little bit of baseline characteristics, they were well matched, and you can see in the 14 patients here, you can see that the median age was about 65. Majority were transfusion independent and a good mix of primary and secondary myelofibrosis, with the majority here with JAK2 mutations, which you might expect, and a mix of intermediate 1, mainly intermediate 2 patients. And then about 8 of these 14 patients had hemoglobins less than 10. So first, let me tell you a little bit about the safety. And so there is a known safety profile with selinexor, particularly in lymphoma and myeloma. And I can say first that there were no new safety signals seen in these patients with myelofibrosis. The majority or the most common adverse effect was GI related, and you can see that at 78% with nausea. Let me delve into that. Firstly, prophylactic antiemetic use is often used with selinexor. Typically today in current treatment, we recommend 2 prophylactic antiemetics. In this study, only 1 was used. And so, and even so, only about 2/3 of those patients received a prophylactic antiemetic. And so the ones that did not, about 100%, most of those patients actually had nausea. Despite that, weight gain was seen by week 24. And so that, in terms of limiting usage, was a transient adverse effect. And as I speak to the current phase 3 study, we have made changes to that protocol to ensure that there are dual antiemetics listed. So the other main side effect or tolerability issue besides GI is actually hematologic with selinexor. And I'll talk a little bit about the hematologic side effects seen with this in this phase 1 portion. 46% of the transfusion independent patients had stable hemoglobin levels throughout week 24. You can see the numbers here, as well as platelet levels that initially decreased but rebounded by week 24. So let me tell you about the efficacy. In terms of, as I mentioned, spleen volume response and total symptom score at 50% reduction in that total symptom score were the 2 co-primary endpoints here in the phase 1. And you can see that in the more conservative portion of patients in the intent to treat, 79% of those patients achieved an SVR35 at week 24. And then in the intention to treat for TSS 50, 58% of these patients achieved a TSS 50 at week 24. All patients in the efficacy evaluable arm achieved an SVR35 at any time on 60 milligrams, and 90% of the patients achieved a TSS 50 at any point in time in the efficacy evaluable portion of the patient population. And more so in terms of some secondary endpoints. So as I mentioned, 79% of patients achieved an SVR35 at week 24. In that data cutoff, as of August of 2023, the median time to that SVR35 was 12 weeks, and 100% in terms of durability of these patients maintained an SVR35 with a mean follow-up time of 32 weeks, and that has actually been extended to about 85 weeks at this point. Similar in the change in TSS, 58% achieved a TSS 50 at week 24, and you can see that the durability was again 100% of those patients maintaining a TSS 50 with a median follow-up time of 51 weeks. We'll say also, we looked at other secondary endpoints in terms of variant allele changes. Cytokine changes at week 4 were associated with a weak SVR at week 24, and the typical 20% decrease in VAF were observed in 5 of the 14 patients, with 3 of these achieving at least a 50% VAF at baseline, and were at additionally high molecular risk patients. So let me lead into the phase. This is the promising data from the phase 1 portion that led into our phase 3 study that is currently ongoing. We are recruiting for 350 JAK inhibitor naive patients that will be randomized in a 2-to-1 fashion. This is a double-blind study of ruxolitinib standard dosing BID with selinexor at 60 milligrams once weekly compared to ruxolitinib standard dose with placebo. And we have co-primary endpoints that have changed a little bit. So again, SVR35 at week 24. And as I mentioned previously in the phase 1 portion, we looked at TSS 50. This is actually an absolute TSS reduction at week 24 with very similar inclusion and exclusion criteria, with which I won't linger because I think I'm running close on time. Let me just end with our phase 2. So in this phase 1 portion, we actually had patients who were on suboptimal doses of ruxolitinib. You can see here at the bottom left at 5 BID, and we had a number of patients who actually achieved an SVR35 at week 24, essentially with which could be considered monotherapy selinexor, as well as achieving total symptom score reductions. This, in addition to an earlier phase 2 study where we saw 4 out of 10 patients achieve an SVR35 in a relapse refractory situation with monotherapy selinexor, led us to develop a phase 2 study of monotherapy selinexor in JAK inhibitor naive patients with moderate thrombocythemia. And you can see the schema here with 2 dose options of 60 as well as a 40 milligram cohort, with the option to add additional JAK inhibitors based on cytopenias. So I think that is my my time, I'm pretty sure. Thank you for again for having me and allowing me to present this promising combination study with ruxolitinib and selinexor.
Yeah, I was just going to say, you know, all we hear about the last several years is combination drug therapy, and it's working, and you know, or at least it appears to be working pretty well. So we're very happy about this drug in the pipeline. I'm very happy to hear you talk about P53 management. That's fantastic, and thank you so very much for being with us today, Travis.
Thank you, Anne.
Thank you. And so we'll move on. It looks like Dr. Agbu is not with us yet. So we're going to move on to Silence Therapeutics and Joe Loers, who's the Senior Director and Medical Science Liaison with Silence. Hi, Joe.
Hi, I'm bringing the video on here.
Okay.
Sorry about that. Looks like it's not working.
Oh dear. Well, we could
Tell
see
you
your,
what,
oh, we could see your face.
you can? Okay. I'm gonna go ahead and share my slides here though.
Now I can't see your face.
All right, let's go back and see. There we are.
There you are. Okay. And you need to share your screen, and I will leave you to do your thing here.
Okay. Can you see a full slide there?
Not yet.
There we
Oh,
go.
there we go. Perfect.
Great. So again, my name is Joe Loris. Thank you, Anne and Mallory, for having us show what's going on here at Silence Therapeutics. I am the Senior Director, Medical Science Liaison with Silence Therapeutics. We're kind of a new company. We've been around since 1994. And I'm going to show you some of the things of where we are and what's moving forward here. But we do have a phase 2 study that we're going to talk about. And let's talk about, first off, and these slides are not going to move forward. Give me 1 second here. Let's see if I can go. I apologize. I might have to go. Second here. So just a few forward-looking statements. This presentation that I'm going to show may contain some forward-looking statements that reflect the company's current views and expectations regarding future events. Forward-looking statements involve risks and uncertainties that could cause actual results to differ materially from those expressed or implied in the forward-looking statements. This is the legal information that we always have to present. And I apologize, I'm not going to be able to slide it into a presentation here, but I will try to make it a little bit bigger. There we go. So Silence Therapeutics, again, our home office, our company headquarters is in London, England. We have 125 employees worldwide. We have a lot of research and development operations in Berlin. And we do have an office here in the United States in New Jersey. So Silence Therapeutics, we silence diseases through precision engineered medicines created with proprietary short interfering RNA technology. So our vision at Silence is to transform people's lives around the world by silencing disease through our precision engineered medicines and driving positive change for the communities around us. Going to talk a little bit about our toolbox here. It considers all elements of short interfering RNA and ligand design. You can see in the first picture here that the short interfering RNA is matched to a target gene. Silence has developed chemical modification patterns that enhance stability and improve activity. Silence has developed a proprietary linkers, enabling the attachment of targeting ligands to the siRNA molecule. Our GynAC ligand delivers molecule to specific liver tissues or cells. It's highly targeted to the liver. So we also have continuous fine-tuning to further improve the performance. So Silence, the siRNA platform and our gene expression, we have a precision engineered medicines. Each siRNA is uniquely designed to only bind to the target messenger RNA, which can reduce the potential for side effects. So it's a highly targeted medicine. There's a broad utility targeting both rare and common genetic diseases. The durability, yet reversibility, siRNA therapies can achieve long-lasting effects after a single dose without permanently altering the gene. In our pipeline, advancing multiple wholly owned clinical programs, we have cardiovascular and rare diseases, plus we have 14 partnered programs. So we have a pretty robust pipeline moving forward. I'm going to talk about Divesiran. This is, we have a phase 2 study going on with Divesiran. We're looking at it in hematologic disorders, and especially polycythemia vera right now for the phase 2. So polycythemia vera is a rare blood cancer with significant unmet needs. And you can see here myeloproliferative neoplasm characterized by the overproduction of blood cells and platelets. An elevated hematocrit is a hallmark of PV disease, indicating overproduction of red blood cells. The treatment goal is to control the hematocrit below 45%. And that's from the 2013 Marchioli study that was published in the New England Journal of Medicine. Also with that same study, patients with hematocrit between 45% and 50% are 4 times more likely to die from CV causes or major thrombotic events than those less than 45%. So it's very important to keep your hematocrit below 45% for a PV patient. PV is considered a rare disease with about 150,000 people in the United States and about 3.5 million worldwide. The current standard of care approaches are inadequate for hematocrit control. Most PV patients are iron deficient at diagnosis. Repeated phlebotomy makes that worse or exacerbates it. So again, the treatment goal is to control the hematocrit less than 45% to reduce CV and major thrombotic events. Divesiran, our product, or SLN124, has a broad therapeutic potential. Divesiran targets the TMPRSS6 and modulates hepcidin, the body's master iron regulator. Positive preclinical data showing therapeutic potential in several hematologic disorders. The proof of mechanism demonstrated was in healthy volunteer study. I'm going to show you a few things about the healthy volunteer study. This data was presented at ASH in 2021, and it was in the American Journal of Hematology in 2023. This study population of 24 healthy volunteers, the efficacy, it showed that the hepcidin increased up to 4-fold and serum iron reduced around 50% after a single dose. The durability effects persisted for at least 2 months during that study period, and the safety and tolerability was well tolerated with no serious or severe treatment emergent adverse events, and this was all in healthy volunteers. Divesiran, clinical rationale for polycythemia vera. There was an article of preclinical data demonstrating hepcidin modulation of polycythemia vera, and it was published in Blood, the Journal of American Society of Hematology. Divesiran elevates your natural hepcidin, leading to the blockade of iron release to the circulation and restriction of supply to the bone marrow. Iron in the bone marrow is essential for red blood cell production, but restriction of this key cofactor reduces the red blood cell generation and reduces your hematocrit as a PV patient. The Divesiran also has 2 FDA designations. We were fast-tracked for polycythemia vera, and we also are an orphan drug designation for PV. Divesiran and how it works. It's being developed for use of people with PV, and the molecule is designed to restrict iron available to the bone marrow for the production of red blood cells. Divesiran increases your natural hepcidin, a hormone that is the gatekeeper of iron in the body. Increases in hepcidin result in a decrease in available iron to the bone marrow, a reduction of red blood cell production, lowering the risk of blood clots and strokes, increasing iron stores to reduce PV symptoms, which include fatigue, brain fog, etc. So I'm proud to announce that the Divesiran Sanrico phase 2 clinical study, it's a phase 1/2 study evaluating Divesiran in adults with polycythemia vera. Phase 1 of the study showed Divesiran to be safe and well tolerated, and I'm proud to announce that phase 2 is now open for enrollment. With that, from the phase 1 study, Divesiran reduces the need for phlebotomies in PV patients. And I'm not sure if you can see all these little red dots on the left-hand side and the orange and yellow here. But these are patients that over a year may have had more than 5 phlebotomies in a year, or they may have had 3 phlebotomies in 6 months. Those patients that are getting continuous phlebotomies, once they came into the screening period and they got their first treatment, you can see that the phlebotomies were reduced significantly. And so that was the phase 1 study. So we're showing that the drug actually has very good efficacy. And this data was presented by Dr. Kremeskaya from Mount Sinai Hospital, and it was presented at ASH in 2024, our phase 1 study. The phase 1/2 study in polycythemia vera, it includes adult patients 18 and over with polycythemia vera, the assessment of safety, tolerability, efficacy, PK and PD response of SLN124 or divesiran. And divesiran is administered by a sub-Q injection. So the phase 1 is completed, and the phase 2 is recruiting right now. So it's a randomized placebo-controlled double-blind study, and the primary outcome measure is the number of patients who are phlebotomy-free after treatment. The phase 2 study aim and objectives is the clinical study is to learn about the safety and efficacy of divesiran in people living with PV. We're looking for about 40 patients that will be enrolled in the phase 2 study globally. The main objectives of the phase 2 study are to assess the safety of whether divesiran works in a broader group of PV patients, the reduction of phlebotomy requirements with divesiran treatment, and the effect of divesiran improving PV-related symptoms, fatigue, brain fog, etc. With this Sanrico phase 2 study, the stages, if you enter the study, there's a pre-screening period. We want you to talk to your hematologist to see if you are suitable to participate in the study. They can provide you with more information about taking part. Then there's a screening process, the assessment of your health to check if it's safe for you as a patient to participate. Then there's the enrollment process. We will work with study centers to make the process as easy as possible. Access to the study centers, there will be compensation for travel. There are no costs for study-related care or study medication. Accommodation will be included where needed. Meals and refreshments are included. Study visit schedule. Visits are every 6 weeks during the study. Assessment, blood sampling, treatment dosing, completion of questionnaires and scales. The length of time that a patient is treated is for nearly 4 years of treatment on this study. Divesiran is safe to date with a long duration of action. Again, safety and tolerability shown in healthy volunteers with patients with PV. There was long duration of action from a single dose, predicting long interval between doses. Sanrico is designed to identify the optimal dose for later development. Polycythemia vera patients may benefit from reduction of phlebotomies during our study and also an increase in iron stores, leading to a reduction in fatigue and brain fog, etc. The Sanrico phase 2 study is now open, and participants can enroll and take part in the USA, Canada, Italy, Germany, Spain, Poland, Bulgaria, the UK, Australia, and Malaysia. If you have any questions of looking for what sites are open here in the United States or around the world, you can go to clinicaltrials.gov and type in Sanrico or divesiran or SLN124, and you'll see the study and the different areas that are participating. Again, if you have any questions or want to look up more information, you can go to our homepage at Silence Therapeutics, silence-therapeutics.com. And I just want to thank you and thank Anne and Mallory for having us. And thank you to all the patients that are about to enter our phase 2 study, the SLN124-04, the Sanrico phase 2 PV study. So back to you, Anne.
Yes, thank you very much, Joe. And what I'm hearing today, which is just wonderful, is the reduction in symptoms, because we get asked constantly, this is great that my blood work is better, and it's great that my spleen has, the reduction in my spleen size, but what about these symptoms that, just never seem to go away? So we're very happy to hear about that, and to decrease and maybe obliterate phlebotomy, that would be wonderful too. So thank you very much for this update. We appreciate it.
Thank you. Bye-bye.
So we'll go back to Merck, who is also a long-term partner, and Dr. Uzor Agbu, who is the Senior Clinical Director. And hello, how are you today?
I'm doing well, Anne. How are you?
I'm
Apologies for the
good.
technical mishap.
No problem. Thank you for joining us, and I will let you take it away, and we will.
There we go. I'll share right now. Okay.
There we go.
Can,
Great. Thank
there we
you.
go. You can see my screen?
Yes, I can see it.
Okay. So again, thanks you for the invitation to talk about Merck and our program today with our agent bomedemstat. I've been, this is, I think, my third year presenting this now. We have a bunch of different updates, but bomedemstat is our license-specific demethylase or LSD1 inhibitor. As Anne said, I'm Uzod Bu, one of the clinical directors at Merck. My main job is to oversee the execution and in particular the clinical aspects of our ongoing clinical trials. So I'll talk about one of the trials I'm running today and those that my colleagues are running. So just some administrative, bomedemstat is investigational. We are still running clinical trials to establish the efficacy and safety of bomedemstat treatment for myeloproliferative neoplasms. And as you'll see from the next slide, we are working across the three classical MPNs, so ET, PV, and MF. I will talk mainly about the ET trials we have ongoing today. That is our most advanced program. But we do have a polycythemia vera trial, a phase 2 trial, which is ongoing. It's not currently recruiting, and we expect to be able to share results from that trial over the coming months. And we also have a myelofibrosis trial, which is completed, and we expect to talk about further steps with that also in the coming months. But going forward, I'll talk mainly about ET. The last time I spoke, we had one phase 3 going on. Since then, we have launched a second phase 3, which would be the first-line phase 3 trial. And then we also have an extension study, which, as you know, most of these trials tend to last a year to 2 years, but this is a lifelong condition. So we want to be able to understand what prolonged bomedemstat treatment is able to do for patients. So our hope is that patients who participate in our trials are able to roll on to the extension study with a far less frequent monitoring schedule, but we'll be able to monitor long-term outcomes when we can begin to look at, do we continue to see progression in symptom reduction? Do we see what is the impact of variant allele frequency reduction? And is there a potential for, is it truly disease modifying? I'll take a step back now and talk a little bit about why we're excited about LSD1 inhibition and bomedemstat. So LSD1 is an epigenetic regulator of the proliferation and differentiation of hematopoietic stem cells. In preclinical models, preclinical MPN models, we see that LSD1 has a number of effects. And the diagram you're seeing here kind of in a simplified way talks about what's happening within MPNs. And in this case, we're looking at ET. So you have the mutated stem cells, which lead to an overproduction of activated megakaryocytes. These elevated megakaryocytes become platelets, and these elevated platelets lead to an increased risk of thrombosis and bleeding. There's also an increase in inflammatory cytokines, which is what leads to a lot of the constitutional symptoms we see within ET. And there's also an increase in growth factors, which eventually leads to increased reticulin and collagen deposition, which causes the bone marrow fibrosis and eventually the need for extramedullary hematopoiesis, which is what leads to splenomegaly. And of course, the mutated stem cells continue to divide and grow, and they begin to become a larger and larger portion of the population of stem cells, which is, as we see, the disease worsens as we go on. Now, inhibition of LSD1 in these preclinical models has shown that it reduces the number of activated megakaryocytes, which would reduce the risk of thrombosis and bleeding. It also reduces the number of inflammatory cytokines, which we would expect to translate to a reduction in constitutional symptoms, so the fatigue, anemia, itching, night sweats, and so on. It also reduces the growth factors and, by extension, reticulin and collagen deposition, thereby reducing the amount of bone marrow fibrosis, reversing that bone marrow fibrosis, and leading to a reduction in splenomegaly. And then key among that, which is our hopes of the disease modifying part that we hope to see with bomedemstat is that it extinguishes the self-renewal of preferentially the mutated hematopoietic stem cells, which would reduce to a reduction in malignant cell population and theoretically should translate into a less severe or disease modification over time. In previous presentations, we've talked a little more extensively about our phase 2 program. So looking at what we saw preclinically, we moved it into the clinic, looking at it in phase 2, where in this case, we treated 73 ET patients. These patients all had at least one prior therapy. Most of them were coming off, about 70% of them were coming off hydroxyurea, but a number of them were coming off of other agents that are used to treat ET. And so they had to be inadequate response or intolerant to at least one prior therapy. In this cohort of patients, you see that 77% of them had a response by week 24, which is achieving your platelets within the target range of less than 400. And these responses were durable. The little figure on the right talks about the durable response, which in this case was defined as a platelet reduction less than 400 for at least 12 weeks. There you see 72% or 52 of the 72 patients included here were able to achieve that. We also saw somewhat of a reduction in symptom burden based on the MPN SAF TSS score. And using the PGIC, which is the Patient Global Impression of Change, 72% of patients reported a favorable response in saying that their symptoms had improved over time, measured from week to week. At week 24, across the different mutations between JAK2, CALR, or MPL, 85% of patients had a decrease in their variant allele frequency. And in the recent publication from 2023, that was presented at ASH by Dr. Gothard, we saw that patients who were homozygous for these mutations, there was a decrease in that homozygosity and they became heterozygous, which theoretically would translate to a less severe illness. Bomedemstat was generally well tolerated with few thrombotic events. As we've discussed in the past, one of the primary adverse events associated with bomedemstat has been dysgeusia. That has tend to be time limited. Some patients have it, but it is generally, some patients have it for a little bit longer period of time, but it's generally low grade and somewhat manageable. The other AEs that we've seen generally tend to be GI symptoms. So there's been some nausea and diarrhea and so on. If you go back and look at the publications for this phase 2 trial, you do see thrombocythemia. That is attributed to understanding how to dose bomedemstat. You can see here, it is a daily oral capsule. You take it once a day. And to sum up, based on this bomedemstat from that phase 2 trial, most of the ET patients that we treated had a clinically relevant hematologic response. Building off of that, we decided to launch our phase 3 program. As I said, last year when we spoke, we had this phase 3 program, our SureSpan 006, which SureSpan is our name for our MPN clinical trials with bomedemstat. That trial has been ongoing for a while, and there we're looking at second-line patients or patients who have an inadequate or intolerant response to hydroxyurea. So obviously you have to have at least one prior therapy. We're looking to enroll about 300 patients. It's in randomized open label trial. So we have 150 patients in each arm. In one arm, you receive bomedemstat at a starting dose of 50, and then that will be titrated to what level it needs to control your platelets. And in the other arm, patients will be able to receive best available therapy, which, as you can see, it would include, because this is a global trial, we have a wide range of things included here, but we have anagrelide, busulfan, ruxolitinib, and the various forms of interferon. For this trial, patients will be treated for about 52 weeks, at which point we'll look at our primary endpoint. And then they'll go on to the extended treatment phase, in which there will be an ability for patients to switch from the BAT to bomedemstat, should we have an indication that bomedemstat is working better than the best available therapy. And as I go through, I don't have it listed here, but for both of our phase 3 trials, the primary endpoint we're looking at is looking at a reduction in platelet count to the target range. We're looking at essentially a normalization of the white blood cell count for those that have an elevated white blood cell count coming in. We're also looking over the course of this 1 year for the absence of thrombotic or major hemorrhagic events. And we're also looking for the absence of progression to MF or AML. Obviously, over the course of the 1 year, that is a little bit rare, but it is one of the things we're measuring for as a primary endpoint. And as Anne alluded to earlier on, one thing that's very important to us is looking at symptoms. So our secondary endpoints deal with a lot of looking at symptoms. Our phase 2 program indicated that we may have a more significant impact on fatigue. So we're measuring fatigue in a number of ways beyond just the in this case, the MF-SAF. We're also using a PROMIS fatigue questionnaire to look at qualitatively what is the impact on fatigue. And we're still looking globally at the total symptom score to see what impact we have overall on symptoms for patients. As I alluded to earlier, we're seeing reductions in VAF with bomedemstat treatment. With this larger population, we're taking a look at what are the changes in VAF over the course of this 1 year of treatment where patients would be on the trial. Again, these are endpoints that apply both to this second-line phase 3 trial and the next first-line phase 3 trial that I will talk about. Before I get into that, I wanted to show you sort of the footprint for this trial. It's a global trial right now with 157 sites. Given the audience I'm speaking to today, I did want to highlight the actively recruiting sites in the US right now. As you can see there, we have sites in California, Colorado, Massachusetts, Michigan, New York, North Carolina, South Carolina, and Virginia. We have new sites opening every day. Well, not every day, but new sites opening up over time. So you can check merckclinicaltrials.com to see if there are sites that are closer to you or in your area if you are interested in participating in this trial. Moving on to the first-line trial, which is slightly different because this is a randomized double-blind phase 3 trial where we're looking at patients who are cytoreductive naive but have an indication for cytoreductive therapy. This indication extends beyond just being high risk. As we know, there are patients who are not high risk who for various reasons need to have cytoreductive therapy, that they're, let's say, intermediate risk patients or those who don't have good symptom control. Their physicians may feel the need to start cytoreductive therapy. In such cases, they would be eligible for this trial. Once again, here, I talked about the primary endpoint. We're looking at a total population of about 300 patients with 150 randomized to each arm. The treatment arm will be bomedemstat again with a 50 milligram starting dose. The control arm here, or the alternate arm, is going to be hydroxyurea. In this case, hydroxyurea is to be administered based on what your physician would normally do. So we're adhering to local practices. However, they would normally prescribe hydroxyurea if you are randomized to this arm is what you would receive. Obviously, it's a double-blind trial, so you won't know what you're on. So there are placebos to mask which one you're on, but in which arm you're on. But patients would be treated in the same manner as they would if they had been just on hydroxyurea normally. Again, this is going on for about 1 year. After 1 year, we expect after the last patient hits 1 year, we'll read out our primary endpoint for this trial. But patients will continue on the trial into the extended treatment phase beyond that 52 weeks. And as I mentioned earlier, hopefully at the end of this trial, roll over to the extension trial. Here, we do not have crossover. We're keeping them separate, so we'll be able to look at a few endpoints that we have over the longer term. And again, there's additional information on this trial at merckclinicaltrials.com. Looking at the footprint, this trial currently has 101 sites. We're obviously getting a lot more than that over time. We currently have 6 active sites in the US, so California, Colorado, North Carolina, Oregon, and Virginia. But as soon as next week, we expect to open 3 or 4 additional sites. So if none of these sites are close to you and you are interested in participating, check in next week again at merckclinicaltrials.com to see which new sites that we've opened and see if one of them would be close to you or feasible for you to participate at that site. And that is all I'd like to share with you today. Once again, thank you, Anne, for the invitation, and thanks to patients and providers who are participating in our clinical trials as we try and work together to advance the MPN field.
This was excellent. Thank you very much. And I'm really happy to hear that the primary endpoints are platelet count, white blood count, and possibly, progression. That's new. That's not out there very often. So we're really excited about this drug. Thank you very, very much. Your list of clinical trial sites reminded me to encourage those that need a second tier list of possible sites to contact us, because we've spent some time researching that. Most people go directly to the main academic institutions and other institutions, and there are many, many MPN docs that would like to participate in trials, as well as patients in other areas. So we really appreciate your time, and thank you so much for this update. It was important. Thank you.
Thank you.
And now we will move on to Cartos and to our friend for a long, long time, Dr. Serge Verstaskovic. It's always great to see you. And of course, you've been kind of a pioneer in bringing other drugs to commercialization. And we're just really happy to see you this morning, or now it's this afternoon. And Dr. Verstaskovic is now the Chief Medical Officer at Cartos, and we will let him explain what's going on there. Thanks, Serge.
Thank you, Anne, so much for the opportunity to connect today. Do you see my screen?
Not yet. I see you.
You don't see my screen here. Just a second.
Do you see a place that says share your screen?
Yes, and it's sharing.
And it's not working.
Just a second. Share, here we go.
There we go. It's coming.
Okay, now we see the screen.
Yes, yes. It's very impressive.
Well, it's impressive, and it's impressive topic that I'm going to talk to you about. Again, thank you, Anne, and your team for allowing me to present on the POIESIS study and the Cartos Therapeutics, the company that is behind POIESIS. As you said, we know each other for a very long period of time. I used to be at MD Anderson Cancer Center in Houston, Texas for almost 25 years, where I developed the single institutional largest center for myeloproliferative neoplasms clinical trials development, and led the development of the ruxolitinib, most recently momelotinib, and participated in development of the other JAK inhibitors. And that actually brings us to the topic of interest. We have JAK inhibitors. They do help patients with the spleen and symptoms, but most of the patients do not derive that optimal control of the spleen and symptoms or the disease features with the JAK inhibitors. And this is where we come in. That's why I joined the Cartos from the academia to help develop the non-JAK inhibitors, the best, in my view, medication that will, in combination with JAK inhibitor with the ruxolitinib, help optimize patient care. And the POIESIS trial has that novel trial design that I'm going to explain. It's underway, and we are very enthusiastic about it. So P53 was mentioned in the past. Cartos is developing investigational agent called naftemadlin, which inhibits protein called MDM2. Now, MDM2 regulates P53, which is depicted on this slide in yellow color. P53 protein goes above, as you can see, and below DNA. So P53 is a clinical tumor suppressor protein that plays an important role in the body's natural defense against cancer. It scans for these abnormalities that you can see on the left side on the DNA. And so normally P53 responds to the presence of cancer by triggering the production of proteins that cause the malignant cells to either stop dividing, which is on the right side, or to die. This is apoptosis. To die is the most important part, of course. However, in myelofibrosis, malignant cells abnormally increase MDM2 levels to prevent P53 from carrying out its usual cancer-fighting functions that is depicted on this particular slide. So here is then the important connection between the p53, MDM2, and naftamadlin. You see in the yellow color to the left, p53 again, tumor suppressor protein, right? Tumor suppressor protein that plays an important role in body defense against cancer. MDM2 in a orange color is the inhibitor of the p53. And in myelofibrosis, cancer cells abnormally increase that protein. MDM2 is very high, preventing p53 from carrying out its usual cancer-fighting function. And then the blue color, the naftamadlin, comes in. That is the drug that we talk about. And you can see to the right, it tightly binds to the MDM2, and MDM2 is then inhibited. We inhibit the inhibitor, and inhibiting the MDM2 restores the ability of p53 to carry its own function to kill the cancer cells. In other words, if you want to make it even simpler, inhibiting the MDM2 takes the brakes off p53, which when activated to high levels, drives myelofibrosis cancer cells to death. That is the apoptosis. And of course, given to the knowing now mechanism described that naftamadlin works in the patients where it needs to activate p53, you will appreciate that naftamadlin activity is restricted to patients whose cancer cells actually have functioning p53. The good news is almost all, 95% of the myelofibrosis patients have a functioning or what we call wild type p53. Now, this is all tied to the biology of the disease, and I want to spend a little bit time on this slide because we actually, you will see in a minute, we actually affect that, all those factors in the patients on naftamadlin. So this slide illustrates what happens in myelofibrosis cancer cells that overexpress MDM2. The MDM2, again, binds to p53, preventing its cancer-fighting ability. And this results in uncontrolled division of cancer cells, as you would expect, and leads to some of the biological clinical hallmarks of the disease, which are depicted in the cartoon here, including increased production of substances called cytokines, pro-inflammatory cytokines that cause many symptoms. Abnormal release of these cytokines results in the disease-related symptoms such as fatigue, night sweating, itchings, bone aches and pains, fevers, and others. The scar tissue that we call fibrosis in the bone marrow impairs the bone marrow ability to produce healthy bone marrow cells, healthy red blood cells, platelets, white cells, and the loss of red blood cells and platelets, for example, leads to anemia and thrombocythemia that again can cause weakness, fatigue, or bleedings. As the bone marrow makes fewer and fewer healthy red blood cells, white cells, platelets, abnormal growth of the abnormal cancer cells may occur outside the bone marrow in organs such as spleen. We know that almost 80 to 90% of patients have a very big spleen. And a liver. Liver is enlarged in about 40% of the patients. And those enlarged organs, again, cause symptoms, abdominal pain, discomfort, feeling early fullness or satiety, decreased appetite. And as we discussed earlier in today's presentations, patients with myelofibrosis are typically prescribed a JAK inhibitor. And ruxolitinib is usually the #1 choice. It does reduce the size of enlarged spleen. It does relieve the symptoms associated with myelofibrosis inflammation. Unfortunately, from my experience, from my publications and others, we know that JAK inhibitors, including ruxolitinib, may not reduce in majority of the patients spleen size or symptoms enough. Most of the patients have a suboptimal control of the spleen and symptoms, not optimal. This is where naftamadlin comes in. We want to optimize the care of the patients with myelofibrosis. With this suboptimal response patients, we know, we believe, and we have results that adding another treatment like naftamadlin, we can actually reduce spleen size further and decrease the symptoms even more, optimizing their care. So what do we know about naftamadlin so far? Naftamadlin has been investigated as both single agent alone and in combination with ruxolitinib in previous studies supported by Cartos in patients with myelofibrosis, and it did show very encouraging activity in terms of reducing spleen size and improving patient symptoms. What makes this drug even more exciting, as I alluded earlier on when I talk about biology of the disease, its potential to change the underlying natural history of the disease to deliver what we call disease-modifying effect. For example, in studies so far, naftamalin conducted to date shown that it can impact the hallmarks of the disease, improvement in bone marrow fibrosis, reduction in a number of myelofibrosis cancer cells in blood, and it can reduce the levels of inflammatory cytokines, all tied to what we call disease modification. In terms of the side effects, this class of drugs, inhibition of the MDM2, are known to cause some irritation of the GI tract or reduction of the blood cell count. However, data from our company, from our studies, from Cartos studies, have shown that these potential side effects can be effectively managed with measures to prevent and treat these side effects. They do not compromise the delivery, optimal delivery of the naftamalin to patients with myelofibrosis. So this is where we come in with the POIESIS. This is the study I would like to discuss with you today. We want to have a name, of course, to help grow awareness of this important study among the global MPN community, including patients and including, of course, our valuable colleagues and friends, our doctors. The study is very important because it looks at the important scientific question, which I alluded to already. We want to optimize the care of the patients that are on a JAK inhibitor and do not benefit enough. But poiesis on its own, as a title, it comes from hematopoiesis, meaning the creation of something that did not previously exist. We know that this is a good name because, as you will see shortly in the next couple of slides, poiesis study design is very unique. We took a different approach than any other company so far. There hasn't been any study quite like this before. It's also an acronym for the mode of action of naftamalin and the clinical benefit we hope to bring to the patients. You see under the poiesis in small letters, P53 activation to optimize responses in myelofibrosis. This is in short, the bottom line, what we do biologically to get the clinical benefit. We have a study website that launched in the United States in February and are waiting for regulatory approval for that website to launch in European Union. And of course, it will be translated in multiple languages for everybody else who may be on a call and it's not within the United States. Now, where are we with the study? Study is already underway, very early in a start, but it's all over the globe. It is in 220 sites across 19 countries, as you can see here. And we launched in June of 2024. As of end of this month, last month, March, we have 140 sites and many more to get to 220 to be activated over the next couple of months. Later on, I will share some further information about the study on clinicaltrials.gov link. And of course, again, I remind everybody that we have active and functional study for that the clinical trial already in the United States. So let's talk about that poiesis study then. The study opened about last summer, takes a patient that have a suboptimal response following treatment with the JAK inhibitor, specifically ruxolitinib. What does this really mean? Suboptimal response means that following treatment with ruxolitinib for a certain amount of time, the spleen remains still enlarged. It's a little bit better, but it's still enlarged. And symptoms related to myelofibrosis are still there, persisting. They may be a little bit better, but they still persist. That is what we call suboptimal response. There are no currently approved therapies in this setting where one would then add another agent on top of ongoing therapy with ruxolitinib to improve the spleen and symptoms to optimize the care of the patients. So study will compare, as you can see at the second bullet, efficacy and safety of adding naftamalin to ruxolitinib, ongoing ruxolitinib therapy, versus adding placebo to ruxolitinib to prove the point that naftamalin is a valuable addition to our armamentarium, first non-JAK2 inhibitor, to help optimize the care of the patients with myelofibrosis. Requirements for participation are usual, 18 years of age or older, have the proper diagnosis of myelofibrosis, and of course, not already on a JAK inhibitor, ruxolitinib, or any other in the past. There are other eligibility criteria that needs to be fulfilled as in any other study. Now, the study design is depicted in part on this slide. There are 2 slides to it. There are 2 parts. The first part here is called, to the left, a run-in period. The second part will be defined a little bit better on the second slide, and it's called add-on period. So focusing to the left on a run-in period, this is where we look for the patients that have not been treated with any JAK inhibitors at all, no ruxolitinib, no any other. So JAK inhibitor treatment naive, as you can see to the left. Those patients are then enrolled into the study and given ruxolitinib monotherapy. That is what we call a run-in period. Ruxolitinib is given for 18 to 24 weeks. The dose is determined by the study doctor. And we then, after 18 to 24 weeks, assess the response to ruxolitinib and determine whether patient has optimal or suboptimal response, or God forbid, the patient has refractory disease. If the patient has a suboptimal response, some improvement in the spleen and some improvement in the symptoms, but not optimal, that is the patient that we will be focusing on in the second part. If the patient is optimally responding or progressing non-responders, they will no longer participate in the study. So patients that have a suboptimal response, right? Some improvement in the spleen, some improvement in the symptoms, but not optimal. They will be assigned to receive either add-on naftamadlin or add-on placebo while continuing ruxolitinib at that dose that they are on that was established during the first part. We are looking for 180 patients that will be randomized, and that is important. 2-to-1 randomization, 120 on naftamadlin, 60 on a placebo. So patients have a 2 out of 3 chance of receiving naftamadlin. And that part is blinded, meaning patients, doctors, or us, the sponsor, we will not know which add-on treatment, naftamadlin or placebo, is being taken. And patients are to remain on the study unless they cannot tolerate treatment or their disease progresses. We will follow patients for as long as we can to also establish not immediate efficacy, as it's usually the goal, but also long-term benefits due to disease modification that I showed you earlier on. This is an important component of the study. In terms of the study procedures, here's the brief review of some of the procedures that patients enrolled on study will need to undertake. That's a standard blood sample collections, the spleen volume measurements, and the symptoms assessment by asking patients to complete a questionnaire in electronic gadgets every day. Procedures will be repeated until week 28 after the randomization in that add-on period so that we can properly assess the contribution of and efficacy, of course, of naftamadlin. The efficacy analysis is done 24 weeks after the start of add-on period, and we will be comparing the 2 arms, naftamadlin or placebo, that was added on to ongoing therapy with ruxolitinib. We will be looking at the spleen volume reduction, as it's common, and improvement in the symptom score in that electronic gadgets that the patient feel all alone. This is very important to do those measurements and will prove the value of naftamadlin to the regulatory body, so we can, with that study, get the naftamadlin to our patients in need across the globe. In terms of additional information, I want to share an important resource that Cartos has developed. That is that website that I mentioned before that has 2 parts. One is for the patients, the other one is for the physicians. There are parts here of interest. For the patients, language is in layman terms, simple, non-technical. Sections include description of myelofibrosis, naftamadlin, trial design, how to qualify for design for the study, and others. And we also, of course, have a trial locations, and which is continuously being updated so that the full information is always up to date. That will be soon enough available for European patients and the colleagues around the globe. In the meantime, there is also clinicaltrials.gov site. There is a number here to the in the right lower corner that one can follow, or email to poiesis.trial@cartosthera.com for further information for us to give you directly. I want to thank you so much for your attention. It was a great pleasure in a short period of time to deliver important news for our community of doctors and patients with myelofibrosis. New medication in phase 3 study for patients in need, suboptimal responders to JAK inhibitors with completely novel trial design. Thank you so much.
Thank you so much, Serge. It was great to see you and great to hear all of this wonderful news. And we do put this information on our website, on our clinical trial page, but also where our industry partners are located on our website. We also can put links in there for our companies that are our partners that will bring them back to this information. But I am personally thrilled to hear all of this great news. You know, it's been a long journey, Serge, as you know. And I mean, it's just, I always say the earth is moving under the MPN community constantly. It never stops. And, but I'm so glad that you're still with us and doing great work. And, you know, you've always been such a great person in the MPN community. We love you. And so but everyone that was with us today, we're just so excited. And many people will watch this. This will be recorded or has been recorded, and many of you and others will be watching. And I'm sure that you'll get a lot of questions hereafter. But thank you very much.
I love you too. Thank you for your
Thanks,
words.
Serge. Bye.
Bye.